Hello everyone! Today we're going to review the differences between the DNA and the RNA. The DNA is a double-stranded molecule and the RNA is a single-stranded molecule. The main difference regarding the sugars, the DNA is going to include the deoxyribose, which, as the name stands, there's an oxygen missing here. And the ribose sugar includes the oxygen. Okay, remember that the name of the ribose determines the name of the nucleic acid. So ribose, RNA, D of deoxyribose, D of DNA. Regarding the bases, the specific base of the DNA is the thymine, and this one includes a CH3 group at this position, while the uracil includes just a CH group. Both of them are going to bind to adenine, and the rest of the bases are the same for both nucleic acids. Now, regarding the replication of the DNA, we've mentioned that the nucleic acids are composed of nucleotides, and each nucleotide is composed of a phosphate, a sugar, and a base. Now the DNA is going to have adenine, guanine, thymine, and cytosine. The DNA is a double-stranded molecule, and when it needs to be replicated, there's going to be an enzyme that it's going to cut the, let me fix just the camera here. Okay, there's going to be a hydrogen bond at this position. Oops, sorry. And there's going to be another hydrogen bond here, and another hydrogen bond here. So between G and C's, we are going to have 3 hydrogen bonds, and between A's and T's, we are going to have 2 hydrogen bonds. This is the reason why A and T base pair and G and C base pair too. So I was telling you that when we want to replicate DNA, so when we want to make a copy of itself, an enzyme is going to cut the hydrogen bonds, and then the DNA is going to add the bases in an anti-parallel way, which means that an adenine is going to be added at this position from a 5 to 3 direction, and a guanine is going to be added here from a 5 to 3 direction. So if we do that, we are going to start with a guanine here, and we are going to add an adenine on the top. So the next one is going to be an adenine here, and we are going to have a guanine here on the top. So if we continue, we are going to have this structure and this structure here. So if you compare both the strands, you'll realize that they are identical. Okay, so this is the replication of the DNA. This is also said to be semi-conservative because for this case, the 2 strands here in the center are going to be the new strands, and the strands in the periphery are going to be the old ones. So semi-conservative means that the half of the genetic material is going to be the original one, and half of the genetic material is going to be the old one. Now, what happens with the RNA? If we want to make a copy of only one of the 2 strands of the DNA, that could be the one here on the left or the one on the right, we are going to separate the 2 strands, obviously, with the previous use of the helicase enzyme that it's going to break the hydrogen bonds, and we are going to start adding the bases. So A binds to T, G binds to C. Now, remember that the DNA has T, but the RNA has U. So A, in this case, is going to bind to U, and the guanine is going to bind to C. So, obviously, depending on the length of the DNA that is going to be copied, it's going to be the length of the RNA, but in this case, I have only 4 nucleotides. So the RNA is going to leave the nucleus, and the 2 strands of DNA are going to base pair. Okay? So far, this was a brief review of the replication of the DNA and the transcription of the RNA. Now remember that for the translation of the protein, there are going to be codons that need to be read in the ribosome, and the anticodon is going to be the corresponding sequence of bases that it's going to be determined, or that it's going to have the correct amino acid, and depending on the order of the codons is going to be the order of the amino acids in the protein. Okay? So as always, if you have any question, let me know in the class.